Viruses. So today we're going to look at how viruses affect a cell. One of the most simple ones to talk about is viruses that affect bacteria, which is what you're seeing here. It's called a bacteriophage. I know it looks kind of like a little spaceship that's going in and injecting material, but it's actually a certain type of virus. We'll go through this and learn that viruses can take on many shapes and sizes. They can affect many different organisms. So that's part of the things we're going to look at. You have fill in the blank notes if you're in class, and if you are learning remotely, then that should be attached to Canvas. So what is a virus? Viruses tend to have a DNA or RNA core and a protein coat. And I'm in the way here. Let's see, move me up a little bit. A biological particle composed of nucleic acid and a protein. Nucleic acid, again, that's what DNA and RNA are made of. And there we go. So again, that other one was showing a bacteriophage, which we'll learn is a virus that affects bacteria. It has a capsid, which is made of protein, and then on the inside it has DNA. And then here's some different types of viruses. I mentioned there'll be several different shapes and everything. Animal virus here has the capsid protein looking almost like a flower shape with the DNA on the inside of that. And this retrovirus is kind of with the capsid or protein all around the outside of it, and then the DNA on the inside there. Intracellular parasites are organisms that must live inside a host, and I have live in quotation marks because a virus isn't technically alive, but that's how it functions. So that brings us to, are viruses alive? They reproduce with the help of the host, so they do reproduce, but not on their own. And that's what is necessary for an organism to be considered living. Sorry, trying to move me again. They have nucleic acid, like DNA or RNA. They adapt to their surroundings. So those are some things that can signify something being alive. Having organization. However, they are not made of cells or cell membranes, which is one of the characteristics of life we learned at the beginning of the year. They can't reproduce on their own. They don't metabolize energy. They don't perform cellular processes. So again, back to virus parts, you have a capsid or a protein coat, and you have some sort of nucleic acid like DNA or RNA on the inside of the virus. So there's the capsid coat of protein that surrounds nucleic acid. Nucleic acid, again, RNA or DNA. Some might have tail fibers, which are used for attachment. They're not legs. There's, again, lots of different virus shapes and sizes. They're all really, really tiny, but they kind of have different shapes to them and can be slightly different in size. There's cowpox, influenza virus. Looks a lot like what you're used to seeing the coronavirus as. Bacteriophage is the one we just looked at before. Those affect bacteria. They look kind of like a spaceship in a way. Herpes simplex virus, HIV-1 AIDS, or the virus that causes AIDS, in other words. Tobacco mosaic virus can affect tobacco plants. Rhinovirus, which causes the common cold in humans. Adenovirus, a respiratory virus. And the Ebola virus here. The shapes will vary again. The virus video is going to be your Amoeba Sisters video, so that will be on Canvas as well. Viral replication. Here's again some of the parts of a bacteriophage listed here. Tail fibers, base plate, sheath, collar, neck, The head is where the capsid is, and on the inside of that would be the DNA. So bacteriophages, viruses that attack bacteria, are often studied. Replication is similar with many animal viruses, and bacteria tend to reproduce several generations much quicker than a lot of other organisms, so it makes them something that's ideal to study. The virus will have two, and in quotations, life cycles, again, because it's not totally considered alive. It functions, but it's not technically alive. The life cycles that they have are the lytic and lysogenic. Sorry, I always want to say lysogenetic. So if you hear me do that about 5,000 times, I'm sorry. It's lysogenic. Step 1, attachment. The virus lands on cell membrane. The virus attaches to a cell receptor. The virus acts as key receptor. Receptor acts as lock, sorry. Virus acts as key, receptor acts as lock. Step 2, entry. The virus enzyme weakens the cell membrane so that DNA or RNA can enter the host cell. Viruses may enter a cell through phagocytosis. If you remember from way back at the beginning of the year, that's where a cell just kind of like surrounds some particle of some sort and engulfs it. Step 3, replication. Virus DNA or RNA uses ribosomes to make virus proteins. Virus proteins created by transcription or translation, so that's kind of what we studied at the end of first semester. So viral DNA is transcribed into viral RNA, and then I keep having to move me. And then the translation of viral RNA into viral proteins. Fourth step, assembly. Virus proteins are assembled into new viruses in the cell. So new viruses get formed in the cell. And then as this happens, the cell, the membrane will get broken down and it will burst. And so the virus enzyme will cause the cell membrane to lyse, which is also bursting. Then the viruses then are released to the rest of the body. In our case, in bacteria, be released to be able to infect other bacteria. And the cycle can then repeat itself. So here's all the stages on one slide. You have attachment, entry of the genetic material, replication of the virus within the cell, and then finally the lysing or bursting of the cell and release of the virus into the surrounding area. Lysogenic cycle. In this, you still have the first step the same as attachment. Second step, entry, is the same as it was before. Third step, replication. Provirus is created where the virus DNA combines with the cell DNA to make the provirus. Infected cell divides by mitosis and copies the provirus. So they're kind of like hitching a ride on the cell's mitosis process here. As mitosis occurs, more and more of these viruses are replicated within the new cells. So each new cell will contain the provirus. Fourth step, assembly. Many new viruses are assembled in many cells. Fifth step, release. Many cells burst, releasing many more viruses. So the viral DNA becomes active and starts making new viral proteins. The infected cells burst, releasing the new viruses. And that is the end of that show. This will again be recapped in your Amoeba Sisters video. Have a nice day.